Hello everyone. Today we'll discuss on the topic gene therapy. So first we need to understand what is gene therapy. So basically gene therapy is the combination of two words, two different words. One is gene. So let me tell you what is gene. Gene is the functional part of DNA which is responsible for any character, like it can produce the protein, and when the gene is used in one therapy, that is called gene therapy is the part of treatment. So basically gene therapy is a technique that is used to replace the defective gene with the new one. So when the gene is faulty, we can replace that with the new one, that is called gene therapy. So it's a very useful technique to treat different type of disease. So basically, there are two different types of gene therapy. One is gene augmentation therapy. So what happens when the gene is non-functional? That means it is not able to produce any type of protein. So in that case, we can replace that faulty gene with the new one. This type of gene therapy is called gene augmentation therapy. But another type of gene therapy is also there, that is called gene inhibition therapy. So in this case, what happens? Although the gene is functional, but it produces the toxic protein, which is not essential for body. So to avoid that toxic effect, one inhibitor is required. So in that case, one new gene can be inserted to inhibit the previous gene, which one is non-functional, or you can say which one is defective. So in that case, this type of gene therapy is called gene inhibition therapy. So the key difference between gene augmentation and gene inhibition therapy is that in case of gene augmentation therapy, the gene is actually non-functional. We need to replace that non-functional gene with the new one. But in case of gene inhibition therapy, although the gene is functional, but it is not the good one. It produces the toxic protein, so which is not required. So to block that gene, we are inserting another gene which can inhibit or mask the effect of previous gene. So that is the difference between gene augmentation and gene inhibition therapy. Coming to the different cell types which are used in gene therapy, so we have two different type of cells. Like one is somatic cell. As you know, somatic cells are general body cells. So these cells can be used to incorporate the new gene. In that case, this is called somatic cell gene therapy. So most of the cases, the somatic cell gene therapy is preferred, and the therapeutic gene is transferred into the somatic cell. So another type of gene therapy is possible, that is called germ-line gene therapy. That means the same procedure, but the difference is that in the germ cell gene therapy, the therapeutic gene is transferred into the germ cell. But the drawback with the germ cell gene therapy is that it can affect the germ line as well as different generation. So to avoid that complication, in generally we avoid the germ cell gene therapy or germline gene therapy. Although it is theoretically possible, but most of the time somatic cell gene therapy is preferred. Then two different type of gene therapies are there, like one is in vivo gene therapy, another one is ex vivo gene therapy. So these are the procedures how we can incorporate the new gene in the cell. So one procedure can be done, that is the direct delivery of gene into the cell of the particular tissue. So here what we are doing, we are just introducing the cell directly into the body. So this type of gene therapy is called in vivo gene therapy. But another method is also there, that is called ex vivo gene therapy. So here what we can do, we can collect the cells and we allow the cells for culture, for growth. So here we can transport the gene into that culture cell. This type of gene therapy is called ex vivo gene therapy, which occurs outside the body. That's why it's called ex vivo gene therapy. So here we need to culture or we need to propagate the cell. Cell culture should be maintained so that we can transport the gene into that culture cell. So that is the difference between in vivo and ex vivo gene therapy. In vivo gene therapy, we don't need any cell culture. We can directly introduce the gene into the patient body. Whereas in case of ex vivo gene therapy, we need to maintain the culture. So this is all about the different types of gene therapy and the basic idea about gene therapy. In our next video, we'll discuss about the detailed procedure of gene therapy. Thank you.